Team serological tests. Plan of the team: antigens of bacteria, viruses, and humans, antigens and autoantigens, heteroantigens. Structure of immunoglobulins, antibodies. Immunoglobulins classes: the interaction between antigen and antibody, types of serological tests, agglutination test, precipitation test, complement fixation test, and immobilization test, neutralization test, ELISA, immunofluorescent test, and polymerase chain reaction mechanism and application. Antigens of bacteria, Escherichia coli, on the left side of this slide, is classified into 150 to 200 serotypes or serogroups based on somatic O antigen and capsular K antigen and febrile F antigen and flagellal H antigen. And on the right side of this slide, you can see antigens of hepatitis B virus. Hepatitis B surface antigen, HBs antigen, and hepatitis B core antigen, HBc antigen, and hepatitis B E antigen, HB antigen, and hepatitis BX antigen, HBX antigen. Next is heteroantigens. Are antigens belonging to different species are called heteroantigens, antigens of plant or animal or microorganisms. An autoantigen is usually a normal protein or complex of proteins and sometimes DNA or RNA that is recognized by the immune system of patients suffering from a specific autoimmune disease. And the structure of immunoglobulins, antibodies. Antibody or immunoglobulin is a specialized glycoprotein produced from activated B cells in response to an antigen, and it is capable of combining with the antigen that triggered its production. And the structure of antibody: an antibody molecule is a Y-shaped heterodimer composed of 4 polypeptide chains, 2 identical light chain, 2 identical this is light chain, 1, 2, and 2 identical heavy chain. This is heavy chains. This is part heavy chain, and all 4 heavy and light chains are bound to each other by disulfide bonds in this area, and by non-covalent interactions such as cell linkage, hydrogen bonds, and hydrophobic bonds. All the chain have 2 ends, an amino terminal end and carboxyl terminal end. There are 5 classes of heavy chains and 2 classes of light chains. The 5 classes of heavy chains are structurally and antigenically distinct. Each is designated by the Greek letters gamma, alpha, and sigma, kappa, and is present only in a particular class of immunoglobulins. And the 5 classes of immunoglobulins: immunoglobulin G, A, M, D, and E, are classified based on the amino acid sequences of the heavy chains, and the light chains are of 2 types, kappa and lambda. And variable region. This is variable regions of immunoglobulins. The first 110 amino acid residues near the amino terminal end of both light and heavy chains constitute the variable regions designated as variable light chain and variable heavy chain respectively. It represents the antigen binding site of the antibody. And the constant region, from heavy chain to light chain. Light chain, constant region consists of light chain and heavy chain. This part. It constitutes the remaining part of an immunoglobulin molecule other than that of variable region. And next is disulfide region. This is disulfide region, yes. And two variable region. If immunoglobulin has two variable region, it is monomer, and immunoglobulin M is pentameric antibody, pentamer, and dimer immunoglobulin A. Function of immunoglobulins. Binding to the antigen is the primary function of an antibody, which can result in protection of the host. The Fab fragment, this is Fab fragment, bears the variable region and is involved in interaction with the antigen. And antiserum, blood serum, that contains specific antibodies against an infective organism or poisonous substance. Antiserums are produced in animals, for example, horse, sheep, ox, and rabbit, and the man is in response to infection, intoxication, or vaccination, and may be used in another individual to confer immunity to a specific disease or to treat bites or stings of venomous animals. And antiserums from animal are most often used, but in response allergic to animals, human antiserums have proved valuable. And the anti-human immunoglobulin G antiserum is produced in gout using purified human immunoglobulin G, and the FC fragment as the immunogenic. And the affinity isolated antibody is obtained from gout anti-human immunoglobulin G antiserum by immunospecific purification, which removes essentially all gout serum proteins, including immunoglobulins, which don't specifically bind to the FC fragment of human immunoglobulin. And monoclonal antibodies are defined as antibodies derived from a single clone of plasma cell, all having the same antigen specificity, produced against a single epitope of an antigen. And when an antigen having multiple epitopes enters the body, each epitope may stimulate one clone of B cells producing one type of antibody, and hence resultant antibody mixture present in serum is said to be polyclonal, and contains mixture of antibodies derived from different clones of B cells. And the principle of this test, a clone of B cell stimulates against a single epitope of antigen, is fused with an immortal cell, myeloma cell, and capable of multiplying identifiably and to produce hybridoma cells, and produces monoclonal antibody of same antigen specificity due to B cell component, and multiplies identically producing clone of identical cells due to immortal myeloma cell component. And types of serological tests, conventional techniques and newer techniques. Conventional techniques: precipitation reaction, agglutination reaction, and complement fixation test neutralization test, and newer techniques is enzyme-linked immunosorbent assay and immunofluorescent assay, radioimmunoassay and Western blot, and immunoassays using electron microscope. Agglutination test. Two types of agglutination tests present on this slide. Slide agglutination test and tube agglutination test. Agglutination test definition: when a particulate or insoluble antigen is mixed with its antibody in the presence of electrolytes at a suitable temperature and pH, and the particles are clumped or agglutinated. Agglutination is more sensitive than precipitation test, and the clumps are better visualized and interpreted than bands or flocculus. Hence, agglutination tests are widely used even in today's modern era of diagnosis. And application of this test, agglutination reactions are classified as direct, indirect, and reverse passive agglutination reaction. All these agglutination tests are performed either on a slide or in tube or in cart or some time in microteter plates. Slide agglutination. It is usually performed to confirm the identification and the serotyping of bacterial colonies grown in culture. It is also the method used for blood grouping. And mechanism of this test: first of all, bacterial colony is mixed with a drop of saline on a slide to form a uniform, smooth, milky white suspension. And next process is to this a drop of serum containing appropriate antibody is added, and the slide is shaken thoroughly for a few seconds. And the next process is a positive result is indicated by visible clamping with clearing of the suspension, or if the milky white suspension remains unchanged, indicates a negative result. And next is tube agglutination. This is quantitative test done for estimating antibody in serum. The antibody titer can be estimated as the highest dilution of the serum, which produces a visible agglutination. And the mechanism of this agglutination procedure of agglutination test: first of all, a fixed volume of a particulate antigen suspension is added to an equal volume of serial dilution of a serum sample containing appropriate antibody in test tubes. And next is positive test, indicates agglutination, clump formation at the bottom of the tube with clearing of the supernatant. Or negative test indicates agglutination has not occurred. Antigen suspension forms a button at the bottom of the tube. And the tube agglutination is used for the serological diagnosis of various diseases, such as typhoid fever. It detects antibodies against both flagellal or H antigen and somatic O antigens of Salmonella typhi. And the H antigen antibody clumps appear as loose, fluffy clumps. O antigen antibody clumps appear as chalky, white, granular, dense deposits. And at acute brucellosis, standard agglutination test we use. And next is precipitation test, ring precipitation test on the left side of the slide, and on the right side of the slide this is precipitation in gel. And the precipitation test, when a soluble antigen reacts with its antibody in the presence of optimal temperature, pH, and electrolytes, it leads to formation of the antigen-antibody complex in the form of insoluble precipitate band when gel-containing medium is used for, or insoluble flocculus or precipitate ring when liquid medium is used. And the ring test this is precipitation in liquid medium. Ring test in a narrow tube. Antigen solution is layered over an antiserum. A precipitate ring appears at the junction of two liquids. Examples of ring tests are streptococcal grouping by Lancefield technique and Ascolis test done for anthrax. And next is precipitation in gel. In other words, immunodiffusion. Incorporation of 1% soft agarose gel in precipitation reaction has many advantages over liquid medium. It results in formation of clearly visible bands instead of flocculus, which can be preserved for longer time. It can differentiate individual antigens from a mixture, as each antigen forms a separate band after reacting with specific antibody. And the immunodiffusion tests are based on two principles, where the only antigen diffuses, single diffusion, or both antigen and antibody diffuse, double diffusion, and whether antigen or antibody diffuses in one dimension, and vertical diffusion when test is done on a tube layered with gel, or two dimensions diffusion in both x and y axis, which occurs when the test is done on a slide or petri dish layered with gel. Yes, this is filter paper strip with Carinobacteria diphtheria antitoxin in the middle of this gel, and non-toxigenic Carinobacteria diphtheria stripe, and unknown toxins, and non-toxigenic Carinobacteria diphtheria. These black lines, this is precipitation lines. Precipitation lines, antigen-antibody complexes. Next is complement fixation test. Complement fixation test, we need sample, of course, and we need complement, and we need coated RBCs coated with its antibodies. And the complement fixation test detects the antibodies in patient serums that are capable of fixing with complements. It was once very popular, now is almost obsolete. And the principle of this test, it is a two-step reaction. Before starting the test, serum inactivation 56 degrees Celsius for 30 minutes, and is done to remove the non-specific inhibitors of complements present in serum. And in the first step, antigen plus test serum plus guinea pig and complement, and as a result, all added together. If the test serum is positive for antibody, next is antigen antibody complex is formed, and the complement gets fixed to the complex, so there will be no free complements in the serum. And if the test serum is negative for antibody, next is there is no antigen antibody complex, and the complements are not fixed, hence remain free in the serum. And in the second step, a hemolysis indicator system is added. It consists of sheep RBCs, sheep RBCs, coated with its antibodies called amboceptors, prepared by injecting sheep RBCs into rabbit. If the test serum is positive for antibody, there is no free complement in serum to bind to amboceptors, and there is no hemolysis. If the test serum is negative for antibody, the free complement are fixed to amboceptor bound on sheep RBCs. Result in hemolysis. Hemolysis, first, if appears hemolysis is negative result. No hemolysis is positive result of complement fixation test. Applications of this complement fixation test. Wassermann test was the most popular complement fixation test used for the diagnosis of syphilis. In addition, complement fixation test was also widely used for detection of complement fixing antibodies in rickettsia, chlamydia, brucella, mycoplasma infections, and some viral infections such as arboviruses and rabies. And next is immobilization test. Treponema pallidum. This is spirochete Treponema pallidum immobilization test is based on the ability of patient's antibody and complement to immobilize the live actively motile Treponema pallidum. Observed under dark ground microscope, it was one of the widely used tests for syphilis in the past, now not in use in the world. Yes, this Treponema pallidum pathogen, and this we add antibody and complement, and Treponema becomes non-motile, immobile. Next is neutralization test. Toxin neutralization test, and the virus neutralization test, two type. Neutralization tests are performed in vitro at cellular level, including cells in the form of organs, and at subcellular level. They assess efficacy of antibody fragments or antibodies to inhibit critical stages of activates, which are generally deleterious to human. These tests mostly employed to viruses and toxins. Toxin neutralization tests are based on the principle that biological action of toxin is neutralized on reacting with specific neutralizing antibodies called antitoxins. Neutralizing antibody defends self from an antigen or infectious body by inhibiting or neutralizing any biological effect. The antibody response is crucial for preventing many viral infections and may also contribute to the resulting resolution of an infection. And neutralization tests are performed in vitro, including cellular level, including cells in the form of organs, and at subcellular level. And neutralization, two types: virus neutralization and toxin neutralization. Toxin neutralization test, this is toxin molecules and plus cell, and cell damaged by toxin. And the next reaction is toxin molecules, and we add antibodies to these toxins, antitoxin, and add cell, and of course antibodies bind with toxins and neutralize toxin and undamaged cell. And the next is red blood cells. We add antibodies, antiviral antibodies from serum, and viruses. And antibodies connect with bind with antibodies bind with viruses, and formation antigen antibody complex is formed. And viruses, viruses, they have one property. Viruses can agglutinate RBCs. And the next is ELISA, enzyme-linked immunosorbent assay. On this slide, you can see types of ELISA: direct ELISA, indirect ELISA, and the sandwich ELISA, and competitive ELISA. Direct ELISA, it is used for detection of antigen in test serum. Here is a primary antibody targeted against the serum antigen is labeled with the enzyme. First step, wells of microtiter plate are empty, not pre-coated with antigen or antibody. And step 2, test serum containing antigen is added into the wall, and antigen becomes attached to the solid phase by passive absorption. Step 3, after washing, the enzyme labeled primary antibodies raised in rabbits are added. And step 4, after washing, a substrate chromogen system is added and color is measured. And next is indirect ELISA. It is used for detection of antibody, or less commonly antigen in serum. It differs from the direct ELISA in that the secondary antibody is labeled with enzyme instead of primary antibody. The secondary antibody is an anti-species antibody, anti-human immunoglobulin, and an antibody targeted to Fc region of any human immunoglobulins. Indirect ELISA for antibody detection. Step 1, the solid phase of the walls of microtiter plates are pre-coated with the antigen. This is antigen. And step 2, test serum containing primary antibody specific to the antigen. This is antibody is added to the wells, and antibody gets attached to the antigen coated on the well. Step 3, after washing, enzyme labeled secondary antibody, anti-human immunoglobulin, is added. This is secondary antibody. And next is step 4, after washing, a substrate chromogen system is added and color is developed. And next, sandwich ELISA. This is it detects the antigen in test serum. It is so named because the antigen gets sandwiched between a captor antibody and a detector antibody. There are two types of sandwich ELISA: direct and indirect, and depending upon whether the detector antibody is a primary antibody, direct, or secondary antibody, indirect. Next, in this third mechanism, this is in indirect sandwich ELISA, the primary antibody and the captor antibody belong to different species, more so the primary antibody is not labeled with enzyme. Another enzyme labeled secondary antibody targeted against the primary antibody is added. Thus, it is more specific than direct sandwich ELISA. Well coated with the captor antigen plus antibody, test serum, and the plus primary antibody plus secondary antibody. with enzyme, and the substrate, we add substrate chromogen. As a result, color is changed. And the next last mechanism is competitive ELISA. Step 1, first of all, I want to say about competitive ELISA is so named because it is antigen in test serum competes with another antigen to same of same type coated on cell on well to bind to the primary antibody. Step 1, primary antibody is first incubated in a solution with a serum sample containing the test antigen. And step 4, this antigen antibody mixture is then added to the microtiter well pre-coated with the same type of antigen. Step 3, the free antibodies bind to the antigen coated on the wall, and the more the test antigens present in the sample, lesser free antibodies will be available to bind to the antigens coated onto well. And step 4, after washing, enzyme conjugated secondary antibody is added. And the last step is step 5. After washing, a substrate chromogen system is added and color is developed. Intensity of the color is inversely proportional to the amount of antigen present in the test serum. On this slide you can see this yellow color, this positive result, and this no color, negative result. Next is immunofluorescent assay. It is a technique similar to ELISA, but differs by some important features. Fluorescent dye is used instead of enzyme for labeling of antibody. It detects cell surface antigens. It is also used to detect antibodies bound to cell surface antigens, unlike ELISA, which detects free antigen or antibody. Principle of this assay: fluorescent refers to absorbing high energy shorter wavelengths ultraviolet light rays by the fluorescent compounds, and it turn emitting visible light rays with a low energy longer wavelengths. The fluorescent dye is used to conjugate the antibody, and such labeled antibody can be used to detect the antigens or antigen-antibody complex on the cell surface. And the fluorescent compounds commonly used: fluorescein isothiocyanate. And the types of immunofluorescent assay: step 1, direct. First is direct immunofluorescent assay. Step 1, sample containing cell carrying surface antigens is smeared on a slide. Yes, this is antigen, pink color. And the step 2, primary antibody specific to the antigen target tagged with fluorescent dye is added. This is antibody with fluorochrome or fluorescent dye. And the step 3, slide is washed to remove the unbound antibodies and then weaved under the fluorescent microscope. And next is indirect immunofluorescence assay. This detects antibodies in sample. Slides smeared with cells carrying known antigens are commercially available. And step 1, test serum containing primary antibody is added to the slide. And step 2, slide is washed to remove the unbound antibodies. A secondary antibody, anti-human antibody conjugated with fluorescent dye is added. And step 3, slide is washed and then viewed under a fluorescent microscope. Applications of this immunofluorescent assay: has various applications such as detection of autoantibodies in autoimmune diseases and detecting microbial antigens, rabies antigen in corneal smear, and detection of viral antigens in cell lines inoculated with the specimens. And last is polymerase chain reaction. Polymerase chain reaction we used for in this slide you can see steps of polymerase chain reaction. First is denaturation, and second is annealing, and third is elongation. Polymerase chain reaction is a technology in molecular biology used to amplify a single or few copies of a piece of DNA to generate millions of copies of DNA. It was developed by Kerry B. Mullis, for which he and Michael Smith were awarded the Nobel Prize in Chemistry in 1993. And the principle of this principle of this method, PCR, involves three basic steps: DNA extraction from the organism. Second is amplification of extracted DNA. Third is gel electrophoresis of amplified product. DNA extraction. First step, DNA extraction from the organism. This involves slices of the organisms and release of the DNA, which may be done by various methods, such as boiling, adding enzymes, such as lysozyme and the proteinase K, and etc. And phenol, chloroform, isomyl alcohol method DNA extraction kits are also available commercially. And second step is amplification of extracted DNA. This is carried out in a special PCR machine called Serological, and the extracted DNA is subject to repeated cycles, 30-35 numbers of amplification, which takes about 4 hours. Each amplification cycle has 3 steps. First, denaturation at 95 degrees Celsius. This involves separation of the dsDNA into 2 separate single strands. And next is primer annealing, 55 degrees Celsius. Primer is a short oligonucleotide complementary to a small sequence of the target DNA, and it anneals to the complementary site on the target ssDNA. And the next step is extension of the primer, 72 degrees Celsius. This step is catalyzed by Taq polymerase enzyme, which keeps on adding the free nucleotides to the growing end of the primer. And the Taq polymerase is a special type of DNA polymerase, isolated from the plant, bacterium, and etc., and capable of withstanding the high temperature of PCR reaction. Last step is gel electrophoresis of amplified product. The amplified DNA is electrophoretically migrated according to their molecular size by performing agarose gel electrophoresis, and the amplified DNA forms clear bands which can be visualized under ultraviolet light. And then amplification of PCR polymerase chain reaction is now a common and often used techniques used in medical diagnostics and research laboratories for a variety of applications. It has the following advantages compared to the conventional culture methods: and more sensitive, more specific, and PCR can be done to amplify the DNA of the organism, either directly from the sample or to confirm the organism grown in culture, and the PCR can also detect the organisms that are highly fastidious or non-cultivable by conventional culture methods. And PCR can be used to detect the genes in the organism responsible for drug resistance. And next is use for primers targeting specific DNA sequence of the organism makes the PCR assay highly specific. And last is amplification. Serological testing is particularly helpful in the diagnosis of rickettsia and viral diseases such as Rocky Mountain and spotted fever, influenza, measles, poliomyelitis, and yellow fever, as well as of infectious mononucleosis and rheumatoid arthritis. As a practical mass screening diagnostic tool, it has proved valuable in the detection of such conditions as syphilis.